Hello. Oh my God, what just happened? I don't know if anyone is still out there, if anyone still has SnapRevise loaded up somewhere. If you do, I've managed to sort of work out some way of fixing this. I don't know if this is a different link. I don't know if I'm here. I don't know if you can see me. Possibly. I don't know. We'll just go for it. Yes. Oh, I can see myself. I'm back. Hooray. Right, guys. Sorry about that. I'll go back to the view that I had a second ago. I'll try sharing my screen with you and see if you can see things. Please comment if you can see this. It might be a bit blurry. I can certainly see something. Logan, you are back. You are here. Thank God for you, Logan. Yes, my followers, my people who are trying to learn biology. I'm so sorry for that. I don't know what happened. My Wi-Fi was totally fine. It just went. It's not blurry for you guys. Okay, cool. Right. Hopefully you can see this. Hopefully you can see what I can see. If I look on my YouTube, yes, you can kind of see the right thing. It looks weird for me, but apparently not for you. Sorry that you now have to go. That sucks. But anyway, where was I? So we were talking about this question, weren't we? And I've obviously lost this now. Oh, is this even the right version? Are you guys going to see a whole load of stuff that you shouldn't see? No, it's not. We're all good. So in terms of this question at the bottom, for those of you that were having a look, so I basically said for this one down here that you need to mention that mitochondria and muscles will need more ATP, and I mentioned that there is a greater surface area if there are more cristae for electron transfer. And one last point, which I think I was just about to say and then I couldn't, was more cristae means more enzymes for photosynthesis, losing it, for respiration. Okay, and then we're going to get to the sort of nuts and bolts of this. Okay, hopefully there are enough of you watching this that this is worthwhile, but if not, there's at least some of you here, so it's worthwhile for you guys. So the first stage of this, the first sort of complicated stage that we need to talk about then, in terms of glycolysis, is we need to take some molecule. So we need to take a hexose sugar most of the time. And when I say hexose, I'm assuming that you guys are aware by now that my hex bit means that it has 6 carbons, as you can see on this diagram. So there's 1, 2, 3, 4, 5, 6. And because it says OSE, anything that ends in OSE in biology tends to mean that it is a sugar. So it is a 6-carbon sugar molecule. Okay, nice and straightforward. The most common one that you guys will ever see is glucose. I guess in your exams, they could try and change this and make it a little bit more complicated and call it fructose or something. But for now, we're just going to be looking at glucose. Okay. This molecule in glycolysis goes from a hexose sugar down to a pyruvate molecule. Okay. In terms of what I mean by that, and I'm now terrified that I'm going to be not here again if I have a look at my account. Let's have a look. Oh, no, looks like we're still here. My pyruvate molecule is basically a 3-carbon compound. So I break it down into a 3-carbon compound. Yeah, please keep messaging me on here, guys, just so I know that everything's still working. A 3-carbon compound. And this process is really important in making a little bit of ATP. Okay, so this process, let's write this somewhere here. This is an anaerobic process. Okay, so it requires 0 oxygen, so it has no oxygen being involved in it. Okay, right, so this is where it starts to get complicated, right? And I mentioned earlier that different specs have different words for different things. So just to show you a bit of an overview of this, this starts off with my hexose sugar. So here is my hexose sugar, and some exam boards like to refer to their one as hexose sugar as opposed to glucose. Don't really know why they do that, but in infinite wisdom, someone decided that was a good idea. Yeah, these are big, scary words, but I promise you it's not too bad. So I've got my glucose molecule up here, right? There is another compound that I know AQA put in here called glucose phosphate. Okay, so there's another one here called glucose phosphate. If you are doing one exam board, you will see that. Okay, there is another molecule that people like to talk about, which is down here. So they like to put GP somewhere down here. If you are doing one of the Edexcels, they like to refer to triose phosphate as GALP, right? If you're doing AQA, this is, AQA, this is just TP. Okay, so be really, really careful with this. Sorry, Charlie, if you've missed 5 minutes. You've missed not too much. You've missed me talking about something, but you can go back in time and see it. This is the main bad boy that we're doing right now. Okay, this is glycolysis. So it starts off with glucose. It gets turned into either glucose phosphate or hexose bisphosphate. And I think you kind of do need to be slightly aware of these names. So you're going to need to probably know about 3 of them. So don't worry too much more than that, but you probably will need to know about 3 of them. Okay. We make something called hexose biphosphate, not bisphosphate. Sometimes you'll see it with a little S here. That is a slightly different molecule. That forms something called triose phosphate, which is also sometimes called GALP. And then that gets turned into something called GP in one of the specs. And then that finally gets called pyruvate. Okay. What I am doing, Christopher, is I'm trying to put all of them together. And I would recommend that you go through the specification for your course and you see which ones they want you to know. Right. So if I try and cover all of them, then you can basically just be like, oh, I'm doing AQA. I don't need to know that one. I don't need to know that one. I don't need to know that. I just need glucose phosphate, triose phosphate, and pyruvate. Okay, so if you just go through and cross out the ones that you do need and the ones that you don't need, that will make your life really straightforward. Okay, so in terms of what's going on then in this sort of stage, I'm going to go through it in 2 sections, and I've handily called this one 1, this one 2, this one 3, and this one 4. So I'm going to stop this slide somewhere along here. Okay, so I'm going to do the first 2 sections here. So the first thing that happens, the first really, really important thing that happens is I need to use some ATP. Now, this is a bit weird because this process of respiration is all about making ATP. So it's bizarre that I actually have to use a little bit of ATP first. Okay, so the first thing that happens is I have to phosphorylate glucose. Okay, this is a term which I mentioned last time. Phosphorylate means add in phosphate to something. Add in, I don't know why I wrote that. Add in phosphate to something. Go away too. You're not wanted here. Right, add in phosphate to something. Okay, so the phosphorylation of glucose basically forms either glucose phosphate or hexose biphosphate, right? I'm going to go for, let's go for this one, just because it says it there. So phosphorylating glucose forms something called hexose biphosphate. And this is not complicated, this step. Essentially what you have done is you have taken, here's these little phosphate groups that you've seen ATP. So here is my adenine, here's my ribose, here are my phosphates. I've literally taken one of these little blue boys and I've just stuck it on to either end of this glucose. So I've stuck one of them here and I've stuck one of them here. You guys don't need to know anything about where it bonds. You don't need to know anything more than that. But the first stage is I need to get some phosphate and I need to stick it to my glucose. Okay. The reason we do this is there's sort of a couple of things you can say. So it either activates glucose, or if you remember this from GCSE, you can say that this process lowers the activation energy required later on. So in any form of reaction, I don't know if you vaguely remember this, you sort of get a graph that looks like this, where you have reactants and products. You have a little line that sort of jumps down like that. The amount of energy needed to start that reaction is this little bit up here, right? So you can say this lowers activation energy. Okay, so lowers activation energy. And let's try and squeeze in down here. Requires 2 times ATP. So it doesn't matter, guys, what board you're on. It doesn't matter which of these molecules you need. You just need to know that this molecule here, glucose, which is always the same, glucose has phosphate added to it, and that forms an activated version of glucose. It forms this version of glucose that can basically do a few more things. Okay, how long will this be roughly? I'm going to try and get through this in, I don't know, add on 5 minutes or 10 minutes to what it would have been normally. So I'm going to try not to bang on for too long. If you need to go, just go at some point and come back and catch up later. Only the true fans are here now. Fatima, you're amazing. I'm glad you guys are here. It went from 40 to 15 watches. Jesus. Yeah, not good. Okay, so the second stage that happens. So I've got my little molecule here of this hexose biphosphate or glucose phosphate or whatever the hell you want to call it. It really doesn't matter because it's all basically the same. And essentially what happens is it will basically just break apart. Okay, you form this molecule which is a bit unstable, and none of the boards need you to know how it happens. It just happens. Okay, so this molecule is hydrolyzed to form two molecules of triose phosphate. So hexose bisphosphate or hexose biphosphate. It looks like someone's forgot to put the S in here. Basically, it's broken in half, and we form two molecules of TP. Okay, so let's say that let's mix things up a bit and make it a little complex. So the activated glucose molecule splits in half, forming two ATPs. What am I on about? Two TPs. Okay, and that's all that's really going on right now. Okay, so I started off with my glucose molecule, I added some phosphate to it, and I turned that into some TPs because it's split in half. Okay, that's all that's going on so far. Nothing more complicated than that. I've just made these two little guys here, and I've started off with glucose. Okay, so add a bit of phosphate on, and you make triose phosphate. Yes, easy peasy so far, right? That is it. So add a bit of phosphate, it activates it. Think of phosphate as being kind of energetic, being an ATP, and then you make triose phosphate. Okay. The second bit is a little bit more complicated, but not a whole lot more complicated, if I'm totally honest. So after we've done this, we need to basically work out a way of getting my triose phosphate to turn into pyruvate. Okay. So we're going to start off by taking some of the hydrogens away from it. So we need to take some hydrogen away from it, which forms reduced NAD or NADH. Okay. How does it split? If I'm totally honest, I think it's just because it's an unstable molecule when it's got these phosphates attached to it, which causes the hydrolysis of it. It could be more complicated. In all likelihood, it probably involves a whole range of enzymes that basically cut it down, but you don't need to know about it. Okay, if you look on Wikipedia at this process, you'll see exactly what I mean. There are literally about 15 little molecules, and each one is slightly broken down a little bit by another enzyme. But don't worry about it. Guys, it's not that hard, Veloki. It's just something you've got to get used to, but it's honestly not too bad. So to turn my triose phosphate, my little activated glucosey thing, into pyruvate, the last stage of this, I basically need to strip off some hydrogen from it. Okay, so I get this molecule called NAD, which is a hydrogen carrier. It is a coenzyme, and it takes a hydrogen off of it, which reduces my NAD to make it into NADH. But in doing so, in taking away hydrogen from my triose phosphate, it turns it into an oxidized state of triose phosphate. Okay, so we need to say for this next stage then, in terms of our little box, that hydrogen gets removed. Hydrogen is removed by NAD. Okay, this forms reduced NAD. So forming there you go. So hydrogen is removed. It's probably going to take an electron along with it, and that's going to then stick to my NAD, forming NADH. Okay, if it's gained an electron, it's reduction. Okay, it's not too bad. Just need to go over it. Yeah, that's exactly it. So yes, this is where this GP comes in. So I think there is an intermediate molecule here, which is basically GP. right? That's where the GP comes in between these two stages. So you'll see that if you do one of the exam boards, okay? I'm not too sure if it's when you take away the ATP or if it's when you take away the NAD. I guess if you think back to photosynthesis, in terms of turning GP into TP, you need to add ATP to it. So I assume it's probably this step here, which is more important in making my GP, but this has to happen too. So triose phosphate is also called GALP, yes. So this little guy here is also called GALP if you do something like EdXL2 or something. I can't actually remember. Okay, I promise you it's exactly the same. It's just got a different name. And if you go on Wikipedia and type in triose phosphate, you'll see that it's called GALP amongst about 10 other things. It's really confusing. It's stupid. And it's a problem with science where if you publish something and you call something a new name, that name sort of sticks. It's really stupid. But anyway, so I need to remove some hydrogen using NAD. So it's oxidized. And then I also have to produce TP by basically taking away these phosphates. So you see these little phosphates that I added earlier. Well, they need to come off again, right? Confusingly, I only added 2 of them, but for some reason, I've got 4 ATP molecules coming off, right? The reason for that is because my little glucose molecule, or not my glucose, one of my hexose biphosphate or bisphosphate or triose phosphates, they basically have a little secret hidden bit of phosphate, which is already there. So production of TP, this has a name. If you add phosphate to something, it's called phosphorylating. If you take phosphate off, it's called dephosphorylating. Okay, so we need to say that either TP or GALP or whatever you call it, TP is dephosphorylated. I can't spell phosphorylated. Ugh. There you go. Is dephosphorylated, forming pyruvate. Okay. Do I do EdXLB? Yes, guys. Aisha, if you meant did I teach it? No, but I think I did it when I did my A levels. If I'm totally honest, it was a really long time ago. AQA is what I've been teaching more recently. Is GP the same as glucose phosphate? No, glucose phosphate is not the same as GP, confusingly. How did GALP turn into GP again? So I think it's when you strip off the when you strip off a phosphate from it. So when you take a phosphate away from triose phosphate, I think that's what turns it into GP. Although I'm not 100% sure on that. It could be when you take away the hydrogen. So I would look it up, okay, just because it's not the same. You don't have to know it for all of you, so I didn't really spend any time looking it up. How did GALP turn into GP again? That's what I just answered. Cool. So that's glycolysis, guys. So you start off, you add some phosphate to some glucose. That forms this kind of molecule, which will have various different names depending on what course you're doing. That will naturally just split into triose phosphate. You need to take off some hydrogen to make reduced NAD, and you need to basically gain some ATP as well. You need to take off some phosphates. Okay. The way I'd think about it, the way I'd try to remember it, is think about what respiration is. Respiration is about ultimately making ATP. So ultimately, we've got to make some ATP for this to be worthwhile. And because you guys have probably done some photosynthesis and you know that hydrogen is really important in chemiosmosis, you know that you've got to be making some reduced NAD wherever possible. Okay, so that's how I tried to remember this. So the products of glycolysis are reduced NAD, NADH, and ATP. Okay, I'm too don't know if I understand that. Okay, I think I do understand that. Right, guys. So in terms of the energy yield that we make, it's important that you understand this. So in terms of ATP, how much ATP did I say that you end up making at the end? Can anyone tell me? Let's get these comments going again before everything breaks and I have to talk to myself for about a minute. How many? Yeah, good. Daniel Chan. So we end up making 4. So we make 4 molecules of ATP. However, the net is 2. Excellent, Fatima. So we make 4. However, 2 were required at the start to activate it. 2 were needed. So 4 minus 2, nice easy maths, equals 2. So my net gain So net being, in terms of, if you've ever come across it in economics, gross is like your total earnings, net is your total earnings after tax, Joy. Your net is 2. So we made 4, 2 were needed, we have a net gain of 2. Okay, why is phosphate added to glucose at the start? It activates it, Aisha. It activates my glucose and basically just reduces some activation energy. In terms of my reduced NAD, you guys need to know that it made 2. So I made 2 molecules of this. Okay, they are used later in oxidative phosphorylation. So I'm going to write that here. So oxidative phosphorylation is the bit which occurs in the cristae. Are they made from GALP? Oh no, someone's getting asked me complicated things. Why is phosphate added to glucose at the start? Are they made from GALP? I don't know if I understand that, Aisha. GALP or TP is just the thing that your glucose, your activated glucose, breaks down into. Okay, good, lovely, guys. I'm loving that you guys are answering this. How many pyruvate molecules? 2 molecules. Good job. Okay, that is because pyruvate is a 3 carbon thing, and glucose had 6 carbons in it. Right, so it breaks down to form 2 3 carbon things. Okay, how are we feeling with this? Post your thing and I'll try and answer your comments. I like there's lots of 2s anyway, because you've said there's 2 pyruvates. Where does reduced NAD come from? Good question, Aisha. Reduced NAD is what NAD is what you mean. NAD is just something that's floating around constantly within the mitochondria. So this is happening in the cytoplasm. So NAD will be floating around in your cytoplasm, and it will basically be just finding any hydrogens that it can. Okay. There are a few 3s here. I'm a little bit upset by that, but that's fine. That happens. People on 3s, right? Key things. I'll go through it really slowly once more for you, because I know there's only like 4 people watching this, and about half of you are saying that. We start off here. So we start off at this stage over here, right, where my little star is. I have a glucose molecule. I add phosphate to it. So I add some of these inorganic phosphate molecules to it, which activates it. As soon as I've activated it, so it turns into this molecule here, I cut it in half. I don't know if I can draw a pair of scissors. There you go, there's a pair of scissors. This molecule will basically be cut in half. It will be hydrolyzed, making my little GALPs, which are these guys, my TPs. Okay, and then to turn that into the last stage, into pyruvate, I need to strip off these phosphates again. So I need to cut them off by dephosphorylating it. So ADP is going to come along and just steal them off. And then I also need to take off some hydrogen as well. So I'm going to take off a little hydrogen molecule from both, making reduced NAD. Okay, that's all that's going on. What I recommend for you guys who are really confused by this is have a re-watch of this in a sec and see what isn't making sense and what is making sense, right? I think if you go through it, it's really not that complicated, okay? Here is an exam question for you to have a go at doing. Here is a question. This is covering the first stage of respiration. See what you guys can tell me, okay? I'll try and draw this again for you. So here is my little glucose molecule. So I'll draw this whilst you guys have a go at going through it, right? And after we've done that, we will go through the answers. So feel free to post your answers. I will have a look, I promise. And I'm just going to draw this for people who are still a bit confused, because I don't like people feeling confused. So here is my glucose molecule, 1, 2, 3, 4, 5, 6. My hexose phosphate is the same molecule, but I've had a little phosphate added to it. I guess the difference here is the same kind of thing. Here's another little phosphate. Triose phosphate has just been cut in half. Right, and then my little molecule down here is, no, it says poo. My little molecule down here is just my pyruvate, and my P has been taken off of it. Okay, right, so first question says, state precisely where this, where in the cell this takes place. Let's see if you guys managed to say this. So have I missed a question? Oh, I have, sorry. Yeah, sorry, I's down here, my bad. So name a stage represented by figure 2.1. You guys have smashed it, glycolysis. Right, well done for working that out. They're not telling you very much. They've told you glucose, hexose phosphate, hexose bisphosphate, triose phosphate, and you got some stuff. Well done, that is spot on. Question 2, state precisely where in the cell this takes place. Zora, lovely. Akib, I'm hoping I'm saying your name wrong, apologies if not. Daniel Chan, Charlie, Decca, Fatima, Mimi, lovely. This is happening in the cytoplasm. Okay, you are spot on. And then finally, identify compounds D, E, and F. This is not ambiguous because they're not talking about the actual molecules here. They're talking about the things that need to go into this. So I'll leave you a few more seconds just so you can all have a go at answering it. Okay, try not to look at each other's answers, but can do if you want to. Give you a few more seconds just because I know there might not be many of you here. Okay, right. Yes, enough of you have said that. So D is ATP, right? ATP needs to go in, just as it has here, to donate a phosphate. If they had another letter coming off here, I don't know, which was like Z, then Z would be ADP. Okay, E, so E's coming down here. Nice, easy one. Look, they've told you that it's reduced. E is NAD. Okay, a mistake that I have seen my students make on this is they have called it NADP, right? Which is very, very, very similar. It's just, it's the one used in photosynthesis. So just get used to the P at the end of NADP standing for photosynthesis. It doesn't actually, but just pretend in your head that it does, right? So this is NAD, that was ATP. Finally, F, lots of you have told me already, this is pyruvate, which is spelt with a U there, despite that looking suspiciously like a V. Okay, so pyruvate would be this last one at the end. Okay, guys, those of you that were really confused, that here is glucose, add phosphate to it, add another phosphate to it, poop. That's maybe a nice way to remember this. Triose phosphate gets split in half, and then you end up taking these phosphates off, donating them back to ATP, and you're left with your little pyruvate. Okay, right, let's carry on. So anaerobic respiration. I'm pretty sure that there's going to be a mistake on this in a second because I had to very quickly load up this PowerPoint and I couldn't delete it. But I'll be intrigued if you guys can spot the mistake. Right? See how eagle-eyed you are. So anaerobic respiration is really, 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 really important. And the reason it is important is because it makes this stuff. Okay, it makes that stuff and it makes a little bit of this stuff. It doesn't make a lot of either of those things, but it makes enough of them. So you've got to think about this in terms of anaerobic respiration being a process that happens when there's not enough oxygen, but it allows you to carry on going. So imagine if you were sprinting away from some horrible organism trying to eat you, right? As soon as you run out of oxygen, which happens when you sprint, you kind of stop breathing, which is weird. If you've ever watched like Usain Bolt run 100 meters, he doesn't really breathe when he runs. It's bizarre. As soon as you run out of oxygen, you need to still have energy, right? And anaerobic respiration allows you to make just a little bit of ATP, so you have a source of energy, and it remakes NAD so this can carry on. Okay, here's a mistake in the diagram, not this diagram, it's in one of the next pages, I believe. Okay, so in the absence of oxygen, half of respiration can't happen. Okay, so I'm going to say only glycolysis can happen. So in the absence of oxygen, only glycolysis can occur. Okay, now the problem with glycolysis, right? The problem with glycolysis, if I go back to, this is nice enough, isn't it? The problem with glycolysis is, let's say I've got a whole load of glucose and I'm turning that into pyruvate down here. The problem with this is all of my NAD, which was here, is being converted into reduced NAD. Okay, that is problematic because if I've only got NADH, then this process will stop because this process requires NAD, right? It requires NAD to strip some hydrogens off of my GALP or my triose phosphate, whatever you call it, and for that to be used later on. So if glycolysis is happening, NAD will become my limiting factor. There won't be enough of it for this to carry on. Okay, the beauty, and I say beauty because I'm a nerd, of anaerobic respiration is in making lactate, which is what we as mammals make, and which is why your legs ache after you've gone for a run, is that in making lactate, I can turn NADH, my reduced NAD, back into my normal NAD, right? The reason it's got a plus here, and I need to be less ambiguous with this, is just because that's another way of calling NAD NAD. It implies that it's oxidized, right? It's had hydrogen taken off of it. So just pretend that little plus isn't there. You don't need to worry about it. Okay. So in terms of why this is important, anaerobic respiration is important because it allows for glycolysis to carry on happening basically indefinitely, right? So buildup of NADH is a problem. It is oxidized. So it has hydrogen taken off of it, which is oxidation, to form NAD. Okay, so if you got an exam question about anaerobic respiration, you need to mention that it allows for glycolysis to carry on happening. Everything else will stop. And the reason that glycolysis can happen is because you are sort of setting your amount of NAD, right? This means that you can make a small amount of ATP. So a small amount of ATP is produced, right? It's just enough to get you going, sort of thing. So what are you guys saying? Let's have a look. Aisha, you are very obsessed with GP. Does GP lose phosphate and turn into pyruvate? Yes. Yeah, you're right. GP is just formed by taking phosphate off of your TP. NAD has to constantly be removed by pyruvate. So yeah, reduced NAD is constantly being removed. So literally you're turning glucose into this molecule here for no other reason than it gives you some NAD. Lactate is really irritating if you are a mammal because it really aches. It's not pleasant to have lactic acid building up in your muscles, right? There's no benefit of it. It just allows you to make some NAD again whilst making a small amount of ATP. Okay. So yes, that is quite clever. It's very, very clever. What's the point in that? What do you mean, what's the point in that? How is NADH oxidized? It has hydrogen taken off of it. So pyruvate will literally just steal a hydrogen atom from it, which forms lactate. Yeah, cramps is exactly what this leads to. Okay, Aisha, try and reword it. So what's the point of NAD? Nice use of NAD. My NADs are important because they basically allow for me to carry on with this process. So if you want to turn glucose into pyruvate, you need some NADs, right? If you want to turn glucose into pyruvate, you need some NADs. If you've turned all of your NADs into reduced NADs, or NADs, then that's a problem. So you need to make NADs to turn glucose into something useful. Okay, so in terms of this reaction, there are sort of two that you need to be kind of aware of, right? If you are a plant or if you are a fungus, then your reaction turns pyruvate. Can you guess what it is based on my little picture? So pyruvate is turned ultimately into, who can tell me? Yes, ethanol. Okay, so pyruvate is turned ultimately into ethanol. If you guys have done any organic chemistry, there is something that you form before ethanol, which is called ethanal, because it's an aldehyde. Don't worry if not, and I don't know if you actually need to know this equation for your exams. I know AQA doesn't need you to know this, but you make something called ethanal, so it's an Al as opposed to an OL. So OL is alcohol, Al is an aldehyde. But you basically turn pyruvate into ethanal. In doing so, F, if you remember your homologous series, you have meth, which means it's got one carbon. F means it has two. So you need to actually strip off a carbon from this molecule. So you release carbon dioxide, which is, if you watched Bake Off yesterday, the reason bread rises in anaerobic respiration. So you need to take off a carbon dioxide. So pyruvate has 3 carbon molecules. Ethanal has 2. And then to finally turn your ethanal into ethanol, you basically need to take off some hydrogen. You need to give it some hydrogen. So my NADH will go in and it will produce a NAD for me, right? So I donate a hydrogen to ethanal, make an ethanol. Okay. If I give it a hydrogen, can anyone spot the mistake that I've made on here? Can anyone spot a mistake, which I actually haven't made on here, someone else made on here, but I'm going to take the blame for it because I should have spotted it myself. What is the mistake on this page? If I add hydrogen to this ethanol, so if I've added hydrogen to ethanol, spot on. This is not oxidized. This should be reduced. Feel free to cross that out on your thing, right? You're right, you could put a plus there as well, person who said that. That's not the obvious mistake. The mistake is in plants and microorganisms such as yeast, the pyruvate it's not oxidized, it's reduced. So this molecule here needs to have hydrogen added to it. If you add hydrogen to something, then that is reduction. Okay, you wouldn't have seen that if this hadn't all broken earlier because I changed it. I just didn't have time to correct it because I wanted to get back to you lovely people. Okay, so the whole point of this, guys, is it makes some NADs. Okay, don't ever forget that. If you ever get a question about this, think about some lovely NADs, right? This is slightly different in mammals. So in mammals, it's a little bit easier. We basically have pyruvate, which gets turned into just lactate. So pyruvate will get turned into lactate. Okay, the way that happens is you essentially are going to have your NADH, which will come off, and it will make a NAD. So hydrogen is going to get stuck to my pyruvate molecule, which basically turns it into something called lactate. Lactate formed. It's broken again. Oh God, don't say that. Ugh, thank you for giving me a heart attack. Yeah, so the benefit of this system is it makes ATP, you make a small amount of ATP, you make a small amount of NAD. So you could, even in this equation, write NAD here, couldn't you? And you could probably write on this equation NADH over here somewhere. You don't need to, but it makes ATP, it makes some NAD. And nicely, if your body basically, if your body has made lactate, the way you get rid of it is you just oxidize it. You add some oxygen to it. And you guys know this. If you did like GCSE PE, this is what I've always heard students saying. If you go into, if you do anaerobic respiration, you go into something called oxygen debt. You don't have enough oxygen, which is why after you've run a race, you carry on breathing really deeply. You get oxygen back in you because lactate plus oxygen turns it back into pyruvate. So lactate plus oxygen will form pyruvate. Okay, so you can then carry on with the rest of the cycle. Okay, is CO2 also a product? I honestly actually can't remember, if I'm totally honest. I can't remember how many carbons are in lactate for the time being. Can someone Google that for me and tell me? So let's have a look. Lactates, I can just Google it myself. I didn't actually think of asking or of doing that. Lactate has 1, 2, 3. No, lactate has 3 carbons in it, so no. In this case, you don't take carbon dioxide off. You don't need to. So lactate has 3 carbons. You just basically need to give it some hydrogen. Yes, it is. Lactic acid and lactate are the same, yeah. Right, guys. So in terms of this, the energy yield is much, much lower in anaerobic respiration. So in this process, you basically make a whole lot less of everything. Okay, so we're talking just about this stuff that's going on over here, right? If I go through this, how much did this make any ATP? How much ATP did I end up making? Can anyone tell me? Including glycolysis. So in this process here, anaerobic respiration, how much ATP do I end up making? And then we'll almost be over. 4, I made +4 over here, but I also had -2 at the start. Where are all these 6s and 4s coming from? 5s, guys. I'm talking about ATP at the moment. 4, 6, 4, 5. I feel like I'm gonna have to go back to the start. Oh, I have to go back to them breaking my computer again. Lovely, Daniel Chan, Decker, you have saved us. So I used 2 at the start, I gained 4 at the end. This process here, look, I haven't mentioned ATP at any point in this process. I haven't mentioned ATP here. This process will make 2 ATPs. Okay, in terms of if I go into my fermentation, which is another word for anaerobic respiration, this will end up making some NAD as well. Okay, so it'll end up making NAD2, which can then feed back into my process over here to turn glucose into pyruvate. Okay, guys, tell me how you think this is going. I know that we've had a break where it broke, but tell me how you think you're feeling with this, and then we're going to call it a day, I think. Glycolysis, for the person who's just asked me, glycolysis is turning glucose into pyruvate, which makes a little bit of energy. So anaerobic respiration doesn't produce any ATP. Glycolysis is technically anaerobic, right? The process at the very, very start is anaerobic. So yes, it does make some ATP, but it's the latter half of it that makes it. So ATP, yeah, sorry, Aisha, I may have confused you with this then. So glycolysis does make a little bit of ATP. It just happens over here. So ATP is made here. No ATP is made specifically in this bit, but all of this is anaerobic. Okay, so anaerobic respiration, glycolysis, it makes 2 ATPs in total. Okay, cool. Right, guys, here is one last little thing, and look, you can see how quickly this, how often this question comes up. I'm glad this is helpful. Have a quick look at this, and then we shall finish. Okay, so where in a cell does stage 1 occur? Who can tell me? So where in a cell does stage 1 occur? Stage 1, glucose to pyruvate. Yes, Fatima. So this is happening in the cytoplasm. Yes, Charlie. Yes, Akib. Yes, Decker. Yes. Ooh. Aisha, no, not the cell membrane. Cell membranes had nothing to do with any of this. I'm intrigued where that's come from. I think you're talking about the cristae, the inner membrane. That is not where this happens. That's where the next stage happens. Okay. All of this is going on in the cytoplasm. Okay, right. Explain how stage 2 enables stage 1 to continue. So how does this guy allow for this to carry on? This is what I've just been talking about. So what can we say? What does it form? Allows reduced NAD to form. Think again, Akib. Charlie Spooks, yes. Or Spokes, sorry, not Spooks. Spooks would be cool though. Stage 2 regenerates NAD, yes. So that was a mistake, Akib, that you made. So stage 2 allows for NAD to regenerate. So it allows for NAD to regenerate. Second mark, what could we say? So it allows for NAD to regenerate. What does NAD do? What was NAD used for? NAD, look back at your notes. What have you said? NAD was used to do what? Turn what into what? NAD is used to make pyruvate. Yeah, can we be more specific than that? So it's used to, let's say, accept hydrogen to form accept hydrogen. Go on, I'll allow that. To accept hydrogen to turn TP or GALP into pyruvate. Okay, that would be your 2 marks on this question. Okay, so it allows NAD to regenerate. That allows TP to be turned back into pyruvate. Where does the hydrogen come from to reduce NAD? So in the previous section, where were we talking about? The hydrogen comes from to reduce NAD. So it's actually stripped off of other molecules. So NAD, you got to think about it as basically being like a thug. It goes around and just takes things from things. So imagine this mug has hydrogen attached to it. If NAD were to come up to it, it would basically rob it of its hydrogens. So yeah, so where does the hydrogen come from to reduce NAD? It comes from my TP. So it'll have a hydrogen attached to it, gets stripped off by NAD, it makes pyruvates. Right, guys. Whew, we made it. I didn't think this would ever happen, but we made it. And well done for sticking with it. A-level king. Love the name, by the way. Just so you know, just so we can actually have a look at these deals I mentioned, coming on the 16th of October, we have SnapRevise 2.0. I'll show you that very quickly. I'll go through it as quickly as I possibly can just to keep everyone happy, including my boss, and to make sure that you guys are seeing everything. Not that, not that, not that. Not that. Oh, I don't have it open because my laptop broke. Right. SnapRevise. Boom. SnapRevise is here. If you go on SnapRevise 2.0, you will see how it works. You will basically see you can pick a subject. It'll give you a test. They give you a load of nice videos which will help you to learn stuff. You can do some quizzes. There are literally thousands. There's a nice little summary of all the content that you can download and keep forever and don't distribute it. You can do some exam questions where basically you'll have an exam question and go through exactly how to do it. There are easy, medium, and hard ones. You'll get some points, kind of like Duolingo or something if you've used that. And then there are people like me who can answer your questions. Okay, that's what SnapRevise 2.0 is. The person who asked me what price it is, if I'm totally honest, I haven't been told everything about it, just because I don't know if it's been totally worked out. If you go onto YouTube and you go on SnapRevise as well, people who possibly haven't seen this before, it's going to play a noise as soon as I do this, so watch out for that. Maybe not. You can see loads of videos that we have made. So there are literally hundreds of these. So here's one which I'm doing right now, apparently. 18 of you are watching. Thank you, guys. You are the amazing ones. Look how many videos there are. Go through them. Like, some of them have had barely any views and they're incredible from what I've seen. So I haven't watched all of them either, if I'm totally honest, but they are really, really good. So have a look through them. There's probably one about what we've done today, which could be useful just for seeing another way of someone saying a similar thing. Basically, with SnapRevise 2.0, the ultimate package is the best one. That's one where you're going to have people like me doing web classes. Whoops, there you go. I feel like Donald Trump doing my little thing on his map. There's going to be something, there'll be the web class on that one, which you can basically see things like this. Okay. Those of you that wanted this little code, there you go, there's the code. You can have 40% off on us, right? This will get you our basic package. It will also get you a discount if you ever wanted to get anything above the basic package, which I would very much recommend. Okay, so over the course of the last 7.5 hours, or that's kind of what it's felt like to me, you should now know what I mean by aerobic respiration. You should know what I mean by glycolysis, and you should know about why pyruvate is reduced and used in anaerobic respiration, and about why it's there, which is all about the NADs, right? If you think about anaerobic respiration, it's all about the NADs. Is there any way of keeping the YouTube live free? If I'm totally honest, I don't know. I doubt it, because I don't think we as a company would make any money if they were always free. I think we'd have to have so many viewers, which you guys could help with, that it just wouldn't work out. Anyway, if you have any questions, now is your time to ask them, okay? I will have a go at responding to you for the next couple of minutes. So, Aisha, if you have any more horrible questions for me, I'll give it a go. But otherwise, I am going to go, and you should go as well. Okay? You have 1 minute, I reckon, and then I'm going to go. I knew it, Aisha. I knew you'd have one question for me. I am waiting. Don't you worry. I'll just sit here and twiddle my thumbs for a bit. I'll drink some more water. Oh, God. Please don't give me a hard question. It's 8:10. I am terrified. Drum roll. God. So GALP turns into GP, which turns into pyruvate, and if so, how? Okay, that question's I'm good with that. So GALP does turn into GP, correct? The way it does that is by taking off ATP. Sorry, is by taking off a phosphate group and donating it to ATP. That is then turned into pyruvate by taking off hydrogen, making NADH. Okay, so do I have a blank page? I've got a bit of a blank page here. So you're saying that I've got TP. You didn't say that. You said GALP, didn't you? Let's make this actually useful for you and not for anyone else who's doing a different exam board. So you said if I've got GALP, that gets turned into GP, which get turned, that is in turn turned into pyruvate. Okay, so you have hydrogen coming off, forming, oh no, you don't, for this first one, you have, no, that one was hydrogen. So this one, you have NADH, DH coming off. So hydrogen is stripped off of it. And this one, you have ATP. So you have a phosphate group being stripped off of it. So ADP goes in, steals its phosphate, forming ATP. For this one, NA, that's a D apparently, not a P. NAD goes on and it takes an H. Let's have a look. Any other questions? Yeah, sorry. My bad. It's been a long day, Aisha. It's been a long day. Happy? I hope so. Right, in which case, I think I'm going to go. If you have any questions, I don't know if any of you remember, in my other one, I think earlier on someone asked me a question, but unfortunately it has now gone because it's in the wrong chat. Oh well, that person can not have a question answer and we'll just ignore him or her, whoever it is. Guys, have a really great rest of your week. If you are still around next week, I will also be around next week. We'll see if we can go a session without anything dying whatsoever. Fingers crossed. Otherwise, it's been really fun. And I will see you then. Let's try and properly stop this from working now. Let's stop you. End stream. See you later, guys. Have a really nice time.